The process of transcription is simply making RNA copies from a DNA template. A few key points. It's not just mRNA. So transcription can also produce tRNA, rRNA, and other non-coding RNAs. Only one strand of DNA serves as a template at a given time. And so you have one strand of DNA, remember it's double-stranded. One strand won't be used for this process of transcription. However, one of those strands of the double strand will serve as a template for the synthesis of RNA. The enzyme responsible is RNA polymerase, and more specifically, RNA polymerase 2, and synthesizes RNA in the 5' to 3' direction. Now, here's a major difference from DNA replication. RNA polymerase does not need primer to start. And so if you go back to DNA replication, you're going to notice that there's an enzyme called primase. And that enzyme called primase actually acts as the starter for DNA polymerase to act. However, RNA polymerase does not need that. RNA polymerase does not need a primer to get started with this work. It does use, however, the ribonucleotide triphosphate, so ATP, GTP, UTP, and CTP as its building blocks to create your RNA strand. What do we need in order to create RNA from a DNA template? One, you need a region of DNA or the gene to be transcribed into RNA. Two, you need monomers for RNA for the new RNA strand. So you need these ribonucleotides in order to create the new RNA strand. Three, you need enzymes to polymerize these monomers. So RNA polymerase is the main enzyme there that you need in order to polymerize the monomers. And polymerize the monomers basically means to help to bond or to create the bond between these monomers. But you also need transcription factors. And transcription factors are needed for gene identification and protein recruitment. Transcription factors are really key in this, and normally when people learn about transcription and they learn about it at a lower level, like maybe, the first time you hear about it in undergrad, you may not hear about the transcription factors also. Their role is necessary. However, most at some levels you focus mainly on just the RNA polymerase 2 and what it does in this process. However, transcription factors are very important not only in understanding, where the promoter is, but also in recruiting RNA polymerase 2, because without a transcription factor, none of this stuff actually happens without the transcription factors. Therein lies another clue when we talk about gene regulation and gene expression regulation. If transcription factors are necessary, they're involved in that transcription regulation as well. So now that we've laid the groundwork, let's look at the overall process of transcription. So genes, meaning specific DNA sequences that code for proteins, are transcribed into pre-mRNA, also known as the primary transcript. This RNA is not functional yet. It still has to undergo processing. So there are three major steps in transcription that we need to know about. One would be our initiation, so the locating of the gene and the starting of RNA synthesis. The other will be elongation. So once we have started the process, it'll be extending the RNA strand. The third will be termination, signaling when this process needs to stop. So initiation, elongation, and termination are key steps in this transcription process. These steps are tightly regulated by specific sequences in the DNA. But even when transcription ends, we're not done. The pre-mRNA must go through some processing steps to become mature, translatable RNA. And we'll talk about that as well. All right, so starting and stopping transcription. Where do we start? So we start at this area called the promoter. So it's a region of DNA that signals the start of RNA synthesis. RNA polymerase and transcription factors recognize and bind there at that promoter region. The terminator is another DNA sequence that signals where transcription should stop. What are the transcription factors? How are they involved? Why do we need transcription factors? We'll talk a little bit about that now. These transcription factors all have different functions, and there's several of these that are key that you're going to have to know exactly what they do. Transcription factor D, so they identify the specific promoter sequences, such as the TATA box, and bind there. Recruitment, so once these transcription factors are bound, they recruit RNA polymerase II and other essential proteins. This illustration shows how multiple transcription factors work together to form the pre-initiation complex. So in initiation, TF2D is the first transcription factor to bind. And the reason it binds first is because on the TF2D, you have a binding site that recognizes the promoter region. It's called the TATA binding protein. So the TBP. The TBP is necessary in order for this whole complex to know where to start the process. It starts the process at the TATA box, but who can identify the TATA box? What transcription factor binds to the TATA box? What transcription factor will get the party started? Well, TF2D gets that party started, and TF2D finds the promoter region by binding to the TATA box. And it does it through the TATA binding protein. So here's how the pre-initiation complex forms. Like we said before, TF2D binds first. It binds through the TATA binding protein. After that happens, then the next step is for TF2B to join. So TF2B binds to that promoter region where TF2D is. Then RNA polymerase II, TF2F, then transcription factor 2E, then transcription factor 2H will bind. TF2H has two parts here where it super matters. One part that really matters is TF2H has a helicase activity. Why would that be important here? Remember, as we are creating RNA, which is single-stranded, we're still having to unwind or unzip double-stranded DNA. TF2H also has kinase activity. What does that mean? Well, TF2H is a protein. It's an enzyme, and as an enzyme, the way it influences other enzymes is by phosphorylating or adding a phosphate group to an enzyme. Adding a phosphate group to an enzyme can do multiple things. In this particular case, it activates RNA polymerase II. And it activates it by freeing it or releasing it from the transcription initiation complex. So upon phosphorylation, RNA polymerase II is released and then allowed to start transcribing. So in this particular case, TF2H is a real key factor here because TF2H plays a helicase function, or has a helicase role, allowing for the unzipping, and TF2H phosphorylates RNA polymerase II, allowing for the start of the process. So transcription doesn't happen without these events. And if transcription doesn't happen without these events, what does that mean? If as a scientist, I want to study, how transcription is regulated, one part I can start is with the essential parts. So if I wanted to see how it really works, take out TF2H and see if the process works. Well, it doesn't. It can't. Because then a few things will happen. One, you won't be able to unwind or unzip the DNA. And two, RNA polymerase II will be stuck to the complex. It's key, right? This means, that's a point of regulation because that part is necessary. TF2D, that's a point of regulation. That part is necessary, right? And so understanding what happens here is a good understanding of gene expression control, understanding what's necessary and what happens to the system if one particular player is missing. So as we talk about elongation, how does RNA polymerase II shift from initiation to elongation? Well, for one, phosphorylation happens. So phosphorylation by TF2H is one thing that really matters. Another thing is activated proteins bound upstream of the gene help promote elongation. So RNA polymerase II as we elongate, RNA polymerase II will add ribonucleotides to the growing strand. We opened up the DNA, we start adding our very first ribonucleotides, and then the RNA polymerase it's riding along the DNA template and adding nucleotides as it goes according to the complementary base pairing. This continues until there's a terminator sequence, and the terminator sequence exists to release the mRNA. Now, finally, before the pre-mRNA can leave the nucleus. And so remember that transcription happens in the nucleus and translation happens in the cytoplasm. But once transcription is done, that transcript must leave the nucleus in order to be translated in the cytoplasm. How does that happen? What happens before it can leave? Well, three things happen before it can leave. One is 5' capping. We talked about this. So as a 5' cap added to the new transcript. That's for stability and for the ribosome to know where to bind. So a 7-methylguanosine is added to the 5' end of the mRNA transcript. That cap is a modified nucleotide, but it's essential for the ribosome to recognize the transcript and then start translation. There's also an addition of a poly A tail that happens at the 3' end. This helps with the nuclear export and protects the mRNA. The poly A polymerase is going to add a series of adenines. It'll be about 50 to 250 adenines to the 3' end of the mRNA transcript. This is called that poly A tail. The poly A tail has multiple roles. It facilitates the nuclear export. It enhances stability. It offers protection from the nucleases. So the longer the poly A tail, the more stable the mRNA can be. So at the 5' end, you have the addition of the cap, and at the end, you have the addition of the poly A tail. But in the middle, we're going to start removing some of the introns. And so splicing removes introns. If you notice here, you have exon 1 here, intron, then exon 2, intron, then exon 3, intron, then exon 4. And so when we finally create a mature mRNA, only these exons are going to be something that we can use to create protein. Those are the only areas here that we can create protein from. These other introns, we can't create protein with. And because we are not creating protein from these introns, we have to get rid of them. Splicing is the way that we go about getting rid of these introns and keeping the coding regions or keeping the exons only. Then your transcript is allowed to leave the nucleus and go into the cytoplasm. And there are sites at the nuclear pore that recognize the splicing, that recognizes the 5' cap, recognizes the poly A tail, and allows the transcript to leave the nucleus and go into the cytoplasm for translation. Thank you so much for following along. Have a phenomenal day. God bless.